And we're going to start. So thanks for joining us today for our last webinar of the series. So good afternoon, everyone, and welcome back to that series. So the series started last year, and we decided to provide it to you again this year on more specific topics. And as I said, that's the last of the year, but hopefully we will keep doing that next year on other specific topics. So keep in mind to put any suggestion about topics that you would like to hear about for next year. And so for those of you who don't know me, I'm Dr. Aude Watrelot, an assistant professor of enology in the Department of Food Science and Human Nutrition at Iowa State University. And I'm co-hosting this webinar with Drew Horton, a research specialist of enology at the University of Minnesota's Grape Breeding and Enology Project. So just before we start the webinar, just few instructions, same as usual, but that just to make sure everybody knows what's going on. So the webinar is being recorded and will be available on YouTube by the end of the year. I will put the link of the recorded webinars on YouTube directly in the chat box during some presentations. Also, if you have any question, we have about half an hour at the end of the webinar for Q&A section. So please type your questions either in the Q&A or the chat box that you can find at the bottom of your screen, where you will see 3 dots. So if you click on those 3 dots, you should be able to type any questions that will be answered at the end. And then also at the end of the webinar, we will ask you to complete a survey if you are willing to do so. It's going to take just like 2 minutes, 5 minutes maximum to complete that. So please take a little bit of time just to complete the survey. So today, that's our great pleasure to have Dr. Peter Salamone. Am I saying your name correctly?
Sure.
Good. From Research Oenovation Collective, and so myself from Iowa State University, who will present practical aspects and research aspects for about half an hour and then half an hour on the use of enological tannin or exogenous tannin in winemaking. So that's just to provide you all the information you may need in your winery. Thanks very much, Pete, for being with us today. And before you will be able to share your screen, I'm just gonna introduce you. So let me give you the privilege. Oh, okay. So you will be able to share your screen while I'm introducing you.
Okay.
So Dr. Peter Salamone is an independent consultant to the alcohol beverage industry. Dr. Salamone has broad experience in industrial biotechnology and the wine industry, both in production settings and E&J Gallo Winery and Diageo, Chateau, and Estate Wines, as well as wine industry service experience with Guzmer Enterprise, Lab4, Enology, Scott Labs, ETS Labs, and enzyme vendors. Dr. Salamone founded the ROC, a nonprofit applied research organization servicing the North American wine industry. Coupled with his diverse production winery exposure, Peter's depth of biotech experience in molecular biosciences, including microbiology, enzymology, and genetics, provides his clients with an outstanding technical resource. So thanks, Pete.
Thank you for that introduction, Aude. I'm having a bit of difficulty getting shared. It's not the same as the one we did yesterday. Let me, clicking on share, and let me get my PowerPoint up and full screen. There. How does that sound?
Looks good. And you sound good too, Peter.
Excellent. Well, very good. Technology is my friend today. First time for everything. Again, thanks to Drew and Aude for inviting me to participate today. I'm certainly glad to be with everybody. We'll just get started right away. A general introduction. Let me just do this. Let me got to go back. Okay, sorry, had a different screen up. So first I'll do just a general introduction to tannins. Then I'll talk a little bit about the use of enological tannins in the winemaking process, different steps, different tannins, what the purposes are, what the proper applications are, including early uses as early as during crush, and then fermentation, later uses during cellaring, and of course finishing. And along the way, I'll stop and mention some specific hybrid varietal challenges, because it certainly does pose a different set of challenges than working with vinifera grapes. So we'll start off with the basics, ABCs. Why are they called tannins? Tannins are a very diverse class of molecules. They're formed from monomers, isoprene units, but the biochemistry is not what we're here for today. But there's a very diverse set of molecules with different structures, and therefore it gives them a very broad range of functions. One of the first functions that was discovered, why they call them tannins, is that the use of these plant-based molecules to treat animal skins rendered the skins preserved in a way that facilitated their use. And so these extracts from plants were termed tannins. The reason they work so well with this application with animal skins is they're able to form combinations with proteins and other vegetal polymers, carbohydrates, polymeric. And so the permanent attachment of the tannins is what gave the property change to the skins, and also what makes a big impact in how tannins operate in wine. Again, tannins are polymers of phenolic monomers. So they're polyphenols. They're usually, they can be monomeric, but of course they become polymeric and larger as they bind with each other. Where do they come from? What is their purpose? Enological, let's just talk about tannins in general. You can see tannins are in just about all plants, and they're in a number of different foodstuffs that we use and in a number of industrial products. They're ubiquitous. They're basically everywhere. There's nothing unique and novel about tannins. Tannins used in winemaking are what I'll call enological tannins. They're extracted from specific sources, and some of these sources are listed in the regulations in both OIV and with TTB as to what's legal to use in winemaking, but they can be extracted from grape, just grape skins and grape seeds. They can be extracted from oak, chestnut, gall, cobraco, exotic woods such as acacia or tara. And so you can see there's a diverse source of these molecules, as well as great diversity within the molecular class. What do they do? What do tannins really do? Why do plants produce tannins in the first place? Well, one of their main properties is they bind to proteins. Grazing animals that eat plants that have tannins in their tissue, it binds with their digestive enzymes. It kind of gives them a stomach ache. So it helps to protect the plant from grazing animals, overgrazing. And also, again, with the binding to proteins, they can inhibit metabolic activities when they get inside of cells and can be detrimental to pathogenic bacteria that plants may be invaded. But as you can see, it's a bit of a plant defense mechanism as to why they make these in the first place. So here comes the chicken wire. No tannin talk would be complete without a couple of pictures of some so-called chicken wire, different classes of the tannin molecules. And you can see that they're basically ring structures. And so the main characteristic, well, let's talk about first, there's two main classes, condensed tannins, okay, and hydrolyzable tannins. Okay, so condensed tannins are these particular structures. You can see catechins and gallocatechins. I'll leave the chemical names off. That's not what's really important for today's talk. But one of the main important things that you want to notice on these condensed tannins is these hydroxyls that stick off all over this molecule that makes them fairly sticky. And that's kind of what their function is. You can see these are monomers. They're a three-ring structure, and they can bind to each other in different orientations to create polymers that have branching and long chains where the rings can stack with each other in orientation and create a great deal of stability, and also create a lot more surface area for the charges to interact with its partners, whether they've been now these molecules don't only interact with proteins, they'll also interact with other carbohydrates, and they interact with a great deal of molecules. And part of the wine matrix, the complexity of it, lends a diverse role to these molecules. The tannins are they're a very strong backbone to your wine structure. So we took a quick look at condensed tannins. Here's hydrolyzable tannins. These are pretty easy to recognize. What you have is a glucose molecule, right? 6 carbons and an oxygen, a 5-member carbon ring with an oxygen. So it's 6-member total ring. And it can be in a straight chain formation when the ring is broken, or it can be in a ring formation. And you can see these gallic acid residues attached to the hydroxyls that come off the sugar, with multiple hydroxyls at the end of each of these gallic acid residues. So you can see a consistency between the hydrolyzable and the condensed tannin in the fact that there's a lot of these hydroxyls that create a lot of electrostatic interactions with binding partners. So what role do tannins play in wine? Well, tannins help with color stabilization. I mentioned the fact that they'll bind with proteins and combine with enzymes and inactivate enzymes. They act as antioxidants. They increase the wine redox potential and protect certain susceptible molecules like flavor aroma compounds from oxidation. They can help to remove reductive characters. Again, that's more the oxidation reduction potential of the molecules. And then I mentioned the backbone of tannins provide for wines, and that creates balance in the wine, balance between sugar, acid, structure. And again, tannins play a big role in consumer and winemaker preference in wines, how they're perceived, whether it's an aesthetic or whether it's a flavor, which we'll get to with finishing tannins and the sources of those and what they can lend to wines. So there's a great deal of impact that the different tannin classes have on wines, and also a great deal of points of intervention in winemaking, where winemaking practices or application of enological tannins can impact your final wine. Okay, when to use enological tannins in winemaking. I mentioned the vineyard. Our talk today isn't really about the vineyard, but there are ways to work in the vineyard to try to help increase the natural levels of tannins. Of course, that's a big challenge with hybrid varietals, is to actually have more tannins produced. They are deficient relative to vinifera varietals in terms of their natural production of tannins. So, but we're going to talk more about enological tannins, and those are tannins that you use in the winery to complement natural levels. And so in the winery, early in the winemaking process, during crushing or pressing, you can use tannins in a sacrificial manner, which I'll explain later. And during fermentation, you can add tannins to stabilize and to build structure in the wines. During cellaring and aging, of course, there's applications for specific enological tannins to add structure to the wine and build that backbone. In finishing the wines, you can add aesthetic and taste, flavor profile to wines. And again, these are specific tannins. And then during transfers and movement, there's actually tannin products that you can add as antioxidants and protection against oxygen pickup and oxidation of wines as they move through pumps and hoses and get placed into tanks. And whenever you do movements, you usually end up with a little bit of oxygen pickup. And in a prophylactic application, you can actually use tannins to help prevent any damage to the wines through that process. So real quick, I'll mention here, you can see with this diversity from the beginning of winemaking all the way through the very end, right up to bottling, tannins are not all the same. What purpose you have for the tannin needs to be matched to the properties of the tannin that you want to use. There's not one tannin that you say, oh, I use this tannin and I use it here, 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 and here. That doesn't work that way. So again, I'll mention real quick now, development of a partnership with your tannin vendor. And there's every company that sells winemaking products has a line of tannins pretty much, and they're not all the same. Different companies have different proprietary blends of their tannin products, and trialing those for your specific need and application is critical in having it work the way that you want it to work. Okay, fermentation tannins. Let's start at the beginning, putting tannins in at the crusher very early on, and for their main purpose to be during fermentation. What does it do during fermentation? Well, tannins bind protein. So soluble protein that gets released when you crush the fruit that can bind native tannins that you want in your wine. You want to keep your grape tannins in your wine. If you add exogenous tannins, they can bind that protein and take the place of the extracted grape skin tannins and preserve those in the wine. I'll talk about that phenomena in detail in a few slides. So it helps to preserve grape tannins. Also, by binding the soluble protein, you reduce the amount of bentonite that you would need to stabilize wines later. If you're making a white wine crushing, well, you can add tannins early on in white wine making as well as in red wine making. Of course, we've mentioned the color stabilization. Anthocyanins, we'll look at structures of those in a bit and how these tannins, specifically condensed tannins, can work with anthocyanins to stabilize color. And we'll also talk specifically about challenges in hybrid varietals with that phenomena. Of course, we mentioned that tannins act as antioxidants. They're very potent antioxidants. They're the most potent antioxidant, stronger than SO2, stronger than other added antioxidants in the wine, and they can help reduce reductive character and also preserve the aromatic freshness by preventing the oxidation of susceptible flavor aroma compounds. Again, we talked about balancing the body and enhancing wine structure and developing the kind of wine structure that is preferred both by winemakers and consumers. And also pyrazine masking and green character is also, tannins can help to mask that effect, that the bell pepper pyrazine character that can come from underripe fruit or in specific varietals. So in fermentation, I mentioned the protein binding and the preservation of native tannins. Let's take a quick look at what, let's say this is a globular protein. You've got a surface area with a lot of different amino acid residues that are presented at the surface, and you've got these tannin molecules. We've looked at the structures of whether they're mostly in grapes and early extraction. You're going to get mostly the condensed tannins. Hydrolyzable tannins are mostly found in oak, so during fermentation, you're not exposed that much unless you add exogenous tannins that have hydrolyzable tannins in them. So your condensed tannins that you have being extracted from your grape skins and some from the seeds, although seed extraction is usually later on when your alcohol levels get a little higher. But you can see that these tannins will bind to the surface of a protein in such a way that will begin to aggregate the proteins and the tannins, and they will precipitate out. So while the proteins are removed, also a good deal of your exogenous tannins are removed as well. That's not what you'd like. You'd like your tannins to stay in your wine. So one of the theories, or one of the practices in terms of adding exogenous tannin, fermentation tannins, would be you add these sacrificial tannins at the crusher, and solubility is important here, a factor to take into account when you're looking at different tannins that you want to add. You want something that's very soluble and able to go right into the juice at crushing so that they're readily available to bind with the soluble proteins. And what they'll do is they'll occupy surface areas on the protein so that as the grape tannins are extracted, there's less area on the protein for the grape tannins to bind, leaving those tannins in solution while the precipitates fall out with some of the grape tannins and some of the sacrificial tannins. So that's how the sacrificial aspect works when you add the tannins very, very early on. Also, the sacrificial tannins are still there, and they can contribute to later tannin interactions that help build structure in the wines. Okay, fermentation tannin effect on bentonite use. I'd mentioned this. Of course, it just goes to follow. If you can precipitate proteins, there's going to be less proteins there later. Bentonite binds proteins, pulls them out of the wine solution, and gives the wines heat stability. And this is just a quick chart. I'm not going to go through details, but as you use tannins in a sacrificial manner, you'll lower the amount of bentonite you need to stabilize wines. Again, you don't bentonite stabilize red wines. This is for white wine applications. And like I said, there are tannins specifically for use in white wines to help with protein precipitation and less bentonite usage later. Okay, let's take a quick look at hybrid issues, talking about protein, cell structure, tannin binding. Hybrid cultivars have less extractable tannin than vinifera varietals. It's one of the challenges that winemakers using hybrid varietals have. They have less condensed tannin. It's just a fact that you have to deal with. Preserving that it becomes a priority. The cell wall, I just put pumice in here because there's it's not exactly defined which molecules in the solids of red hybrid varietals do the binding, but they do bind quite a bit of tannin, maybe even more so than vinifera varietals. It's been very difficult to quantify that effect in terms of specific molecular interactions, so that knowledge is not readily available. But it is the general phenomena of the tannin binding has been identified and quantified, but the specific interactions have not been identified to this point, to my knowledge. Okay, So hybrid cultivars have significant levels of soluble protein. When you crush the fruit, there's a significant amount. Measuring it can be difficult. I won't go into the details of this, but the high sugar content makes many of the standard protein assays non-functional. But there is an amido black assay that I know I've worked with Larry Brooks before and looking at in musts early on, measuring protein amounts. In the amido black assay, you can actually do that with. But they have significant levels of soluble protein that also will challenge your tannins in solution. And then copigmentation, I'm going to make it real simple, and this isn't a complete definition, but copigmentation is a non-covalent association of molecules with anthocyanins. Of course, anthocyanins are your colored compounds, and the non-covalent associations occur pretty rapidly early in fermentation. But the problem with copigmentation is that those associations deteriorate as alcohol levels increase. And then the stability of your anthocyanins becomes dependent upon whether they were polymerized into oligomeric size polyphenols, because after these copigmentation associations deteriorate, monomeric anthocyanins are very unstable. Okay, we'll talk about more about that when we talk about anthocyanins, but these are some of the issues in hybrid winemaking early on with tannin use. Okay, so approaches in the cellar, what can you do to help meet some of these challenges? Early tannin addition, again, right at the crusher, get it in there early to bind with proteins and maybe even occupy some spaces in the pumice, in the solids, where your grape tannins may bind and then end up not in your wine. So the early sacrificial effect, it can't hurt. The tannin levels are already low in hybrids, so to preserve what you can out of the fruit itself is probably a really good strategy. Also, like I said, some of the early tannin addition sacrificial tannin remains in solution and can become part of that polymerization that you desire to build that backbone. Sequential aging tannin additions throughout cellaring. Again, you add an early tannin, you're not done. You're starting off at a deficit. You need to build some more structure in your wine. Tannins, you can't just dump them in one big batch and they do what they do and you're good to go. They react slowly. They react with each other and with other partners. The reaction matrix changes as the ethanol level increases. And so you've got to tease them in a little at a time through multiple additions. So keep that in mind. To use enological tannins to build that kind of structure in your wines is a commitment. And each step of the way, it's going to take some trial and error. There's not a recipe. Winemaking is not formulaic like brewing. And so, you're going to have to do a little trial and error, not only at timing of addition, but also the conditions in your cellar and also which tannin product you're adding, because they're different. Okay, so, and then here's just an experimental. I'm just throwing this out there. This is me putting some spaghetti against the wall. Okay, if you've got issues with proteins, an addition of a proteinase could change the structure of that protein in such a way that maybe it doesn't bind tannins as well. Okay, or it can change the structure of the protein such that they bind each other more strongly and leave more tannins in solution. So that's one way proteinases could be used, there's other carbohydrases, arabinases. These are enzymes that cleave arabinose residues off polymeric carbohydrate chains. Same with galactosidase and galactose. These modify. Let's just digress real quick here. When you crush fruit, when you crush grapes, pectin is a major component, and pectin breaks down. Even if you don't add pectinase enzymes, there are native pectinase enzymes that over time break that native pectin down in the fruit. And fragments of this pectin are large carbohydrate polymers that exist in your wine. They bind with tannins, they bind with proteins, they bind with anthocyanins. They're there the whole time. And that's when people say colloidal structure, generally what that is the functioning of these pectic fragments that are still in there associating with these other compounds in the wines. Okay. Now, a lot of these pectic fragments have arabinose and galactose residues. So this can change that and can also help with the binding and shielding and preservation of things like tannins and anthocyanins. Okay, so again, and then I mentioned beta-glucosidase with an asterisk. I'm going to talk specifically about use of this enzyme in again, a spaghetti against the wall experimental approach to looking at preservation of color in hybrid wines. Okay, we talked about sacrificial tannins, and I didn't mention any product names. I don't work for a company anymore. I don't advocate for any company at all. But because of the challenges inherent, we're talking about building tannic structure. There is one product that I want to mention, and I'm only mentioning this not as a pitch for the company, but because this product is patented. So it's only one company's product, and it does a very unique job. This is a tannin VR color. It's a Lafort fermentation tannin, and it's very good with color stabilization. It's a natural active catechin, which is a condensed tannin, and the way it's extracted and produced leaves it in a very reactive form. It forms covalent bonds between tannins and anthocyanins through a acetaldehyde bridge, which I'll go into in a second here. And what it does is it helps to preserve the tannin potential of the grapes because it binds up the native tannins with the reactivity of itself as it builds polymers. And another factor of this is that it has very low astringency. So you can add this tannin and it doesn't add any bite to the wine at all. It doesn't have gallic acid. And just the way that it acts by being very good at polymerization, it just doesn't bring astringency when you add it. It's usually added at a maximum of 300 ppm. I've done experiments where I've added it at higher levels. I've never seen it do any more than it did at 300 ppm. So there's really no need to add more than that, I don't believe. Okay, let's take a quick look at what this does chemically that makes it unique. You have in wine, you have early in fermentation. It's a bit still of an oxidative environment. The yeast likes some oxygen early on. There's oxygen in the must from the crushing and all. And early ethanol can be oxidized to ethanal and then acetaldehyde. This is acetaldehyde. This is a molecule that works with reactive catechins to form a bridge through this particular bond that I'm highlighting here. And then it can become a carbocation, which is extremely reactive, and it reacts with anthocyanins and other catechins or condensed tannins in solution to begin this polymerization process through this acetaldehyde bridge. Now, in this anthocyanin, I'll point out right here, this oxygen, this is your color center. This is the guy that takes in wavelengths and re-emits wavelengths, and depending on the subgroups of this anthocyanin, these R groups can be different. The color from red to orange to purple is pH dependent and also dependent upon the chemical environment around this oxygen. So you can see that by building this bridge with this reactive catechin, that it protects this center and stabilizes the color, stabilizing not only in terms of keeping this from precipitating, but also stabilizing the chemical environment around this oxygen and keeping the color stable. Okay, so I mentioned this was a unique product. I'm going to show a quick reactivity. This is a benchmarking study with a number of fermentation tannins. You can see them all down here, and they all have a bit of reactivity. This is a reactivity assay where these were added with molecules, and they were measured how rapidly these things would bind and polymerize with partner molecules. As you can see, most of these are down here around under 10. Okay, then you've got a few of these that are 50, 59, 170. These are grape tannins, very reactive, good. This is why you want to keep your grape tannins in solution through a sacrificial effect. These are good tannins. They react fairly well, but you can see that the VR color reacts at over 5 times the next highest one. Another point I'll make is that these tannins, these reactive grape tannins, these things are very expensive. They're like 300. I can't quite quote prices anymore. It's been a while since I've been on the commercial side of the industry, but they're like $300 a kilo. This is the same price as a regular fermentation tannin, maybe like $35 a kilo. So you can see you get a lot of reactivity, more than almost any other product. So, all right, enough about that. The reactivity is important though. I'll talk a little bit, let me go back here again, just with reactivity. I'll mention briefly too, when I talk about hybrid varietals and some of the work that was done that I use in this talk today, is that the reactivities, they're not non-reactive, they're just slower, okay? And time is not your friend when it comes to the timelines of tannin reactivities in winemaking. You have, maybe a year in the cellar, but a lot of times you need to preserve some of these molecules in polymeric forms earlier, or the loss of them through precipitation and destabilization is much more rapid than the polymerization reaction. So then again, it gets to be a time question and not just a reactive, not reactive question. Okay, I mentioned it has very low astringency. I'm not going to go into details on this, just to say that, you know, I say that it's measured in a gelatin binding assay, as well as through aesthetic taste comparisons. Fermentation tannins, when to use them, how to use them. They're protein reactive tannins. You want to put them in very early if possible. After crushing, so that, again, we talked about the protein binding, the binding to in spots on the solids that would otherwise take up your grape tannins. You want to try to maximize your grape tannin retention through the process. So adding these early on, and this is this rate for hybrid varietals is really much higher. Some of the experimentation that's been done has been up as high as like 900 ppm. So. But when you add this, like I mentioned, a supplement, this doesn't replace these protein reactive tannins, the VR color, the reactive tannin. It doesn't replace these. So you should use them in conjunction with these, okay? And the time to add VR color is a little bit later. Like I mentioned, you want some oxygen still in your ferment. You want a little bit of oxidative environment to help the acetaldehyde formation and the reactivity. So you need a little bit of ethanol produced. Okay, and you need to allow that oxidation of that and the acetaldehyde production to become a part of the wine matrix. So about a drop of 5 or 6 bricks, you know, maybe a little less than a third of the way through the fermentation, is a good time to do your VR color add. And like I said, I've never seen it need more than 300 parts. Okay, I want to talk about real quick with fermentation tannin and additions during fermentation, and then the effect throughout post-fermentation in the cellar. I'm not going to go through the details of this study, but it's great just to take a look at this and understand that post-fermentation, preference of different tannin additions or no tannin additions, or it's all over the place, okay? It starts in one place, it goes another place. It's just, for the first 6 months, wines mature differently based on what was added in them. So if you do a fermentation add, okay, and then you do an early cellaring add because you want more tannin in to help with your structure, and then you want to do maybe a second one after 3 months, maybe you do a little bit of wine movement or microoxygenation to help with the phenolic polymerization reactions, okay? You've got to give it about 6 months. After 6 months, the wines begin to take on a character that they maintain. And if you look at those characters that are maintained after 12 months, post additions, okay, then you start to see that the tannin additions are preferred over oak chips alone or no additions at all. So some people say, I'll just use oak chips instead of adding tannins. I get the same result. Not really. Oak adds some flavor that some tannin preparations don't, and that certainly can be a benefit. But overall, fermentation tannin additions are preferred in the long run, but you have to give them time to mature. Things change a lot in the cellar. Okay, let's take a closer look here at hybrid issues with anthocyanin structure and reactivity. We're going to take a quick look at what molecules are present, what form are they in, why does it matter about the form, and what can we do about it? Okay.
You're going to have about 5 minutes.
Okay, very good. Like I said, I definitely won't do the case study. Okay, real quick, from Birch and Mansfield's publication, you can see that in hybrid varietals, there's, you know, different forms of these anthocyanins, okay? And they're all in the diglucoside form, 3,5-diglucoside form, and they have different colors, okay? Across different varietals, in this particular instance, Frontenac, Marquette, Saint Croix, you can see that there are different levels of anthocyanins. Now, within this anthocyanins in these varietals, you can see that there's a proportion of diglucosides to monoglucosides. There's many more diglucosides. In vinifera, okay, there's very few diglucosides, and they're mostly monoglucosides. That's a big difference when we're looking at things like this, okay? Here's your anthocyanin. Remember the oxygen that is the color center. There it is. Here's a glucose molecule substituted onto this. This is looks like a condensed tannin, but it's an anthocyanin, and now it's stabilized. It's got a glucose. That's monoglucoside. That's a good molecule. Here's a color molecule, same exact color center, everything. Here's the monoglucoside all right? And then you've got a diglucoside. You've got a second glucose molecule added, all right? Now, this helps to stabilize the molecule, but it challenges hybrid varietals in the formation of polymers. It's much more difficult through steric impedance and removal of this as a potential reaction partner for this molecule to get incorporated into polyphenols and to stabilize these anthocyanins long term. That's a big challenge, okay? So with that kind of a challenge in mind, let's take a quick peek here at an experimental approach. This is me throwing spaghetti against the wall, and since I'm kind of low on time, I'm going to run through this really quick. If I was doing an experiment with hybrid varietals today with the tools that I think are available and with the challenges I know, here's some of the things I would do. I would think about an application of a beta-glucosidase. This is an enzyme that cleaves glucose molecules off of other things, okay? They're readily available. They're used in mature aroma compounds in finished wines. They have different substrate specificities. Finding one that may work on a diglucoside anthocyanin is not out of the question. They can be promiscuous and highly active in a wine matrix, including 14% ethanol. They work there. You can stop the activity by bentonite if you want to halt it. They're inhibited by free glucose, so the timing of addition is important. You can't add them early. They'll be inhibited by the sugars. The reactive tannins that you need, the acetaldehyde, this would be like a VR color thing, to form the bridges between molecules, okay? Acetaldehyde is a thing that happens early in the wine, but it can also be a byproduct of yeast metabolism. It can be later, produced later as well, so it could be there, or you could even add some. And then I mentioned that enzyme application with microoxygenation after fermentation, when the sugars are gone, microox plus the enzyme develop acetaldehyde bridges after we've glucosidase treated the anthocyanins to try to get them in a more reactive state to match up with your activity of our tannin, okay? So here's a word of caution. Deglucosylation of anthocyanins is generally thought to destabilize them in wine. But stability isn't only about getting into polymers, it's also the stabilization of that color center electronically, like I'd mentioned. So deglycosylation and destabilizing color isn't necessarily meaning that you're not producing polymers, but you're exposing that center to pH and other effects that could change it. Okay, real quick, through the finishing tannins. Again, all right, this is a very diverse group of products. They're made from untoasted oak, from toasted oak, from other wood extracts. You can see by the pictures, they come in various colors because of the toast and the extraction and the source and everything are very different. I'm going to show you some benchmarking data. I'm going to whip through it pretty quick. I'm going to show you some benchmarking data that was generated in the past that I was never able to show, but I can show it now. There's a lot of different products mentioned, a lot of different companies. I'm not advocating for any one over the other. What I want to show with this data is that there's differences. Okay, what are we going to look at? Physical aspects. The solubility, important. Is it going to remain insoluble? It's not going to help your wine. The turbidity, okay, is it going to make your wine cloudy? That's not good. Filterability, of course, you don't want to add this to your end wine right before you go to bottling and plug a filter. That's going to suck. Okay, color intensity. You can see they're different colors. Are they going to bring a color you don't want to your wine? Then aesthetic aspects of these products. Astringency, there's a gelatin index. Do they bring astringency in or not? And then with oak compounds, there's different flavors and tastes that can come from these toasted oak extract products. Here's a list of the products that were tested, and now this is being recorded, so this is all available. You can look it up later and take a look through some of these to pick somebody that you may want to contact about their products. Okay, turbidity. The point of this graph isn't who's good and who's bad. It's that they're different. Okay, and I don't even know with 50 NTU, is that going to create a problem at the filter? I don't know. Are you doing 0.8 filtration? Are you doing 0.4? Are you doing absolute 0.2? Are you how are you filtering your wine? So, and maybe you'll change that. If this makes a wine that you just love, maybe you want to do an unfined, unfiltered post this tannin addition. So there's lots of ways to address things. Filterability, again, most are around reasonable levels, and a couple of them can have some issues with filterability. Color intensity, does it change the color of the wine once you add it? Again, it's a continuum from high down to low. So each product is a little bit different. You've got to trial it in your wine. And this is a great bench trials with multiple glasses, with a number of products and working. This is what is usually the fun part of winemaking. fine tuning and correcting. Again, the addition of flavors or compounds from, say, the oak, toasted and untoasted oaks, whether it's vanillin or whiskey lactones or furfural, you can see there's differences in these products. They're not all the same. One supplier is not the same as the other. So whiskey lactone, again, another very positive attribute that most people would really, that's why you put your wine in oak a lot of times. And then, of course, at the end of the day, aesthetic aspects, the tasting profiles. What do these things bring in terms of tasting? For all the way down from like very flavored up to more of a roasted effect. And then this is intensity of the aesthetic. So, and again, another big difference, makes a difference, the price. Some of these are super expensive, and you have to really want it for your wine because it's going to add pennies to your cogs, okay? But some of them, most of them are fairly reasonable, and certainly for what it brings to the table, it's usually a pretty good investment. All right, I want to acknowledge the sources that I used for some of the information, along with, I also want to mention, I've been in the industry for 20 years. I've been on the production side with Gallo and Diageo. I've been on the producer or on the supplier side with Lafort and Gusmer Enterprises. And then again, I've been on the winemakers side with the Research Oenovation Collective and helping through experimental work to bring more knowledge to the industry. But, these were resources I used for this particular presentation. So we did a general introduction, talked about enological tannins in the process, and mentioned a few specific hybrid varietal challenges and some approaches to overcoming those. Thank you for your time and attention today.
Thanks, Pete. I saw that we have already one question in the chat box, but stay with us. We will answer that question at the end of everything.
Very good.
Pete, thank you for that presentation. That was a really deep dive in a little under 40 minutes. I could imagine a full college course on everything you just stuffed in there. We appreciate it. I'm glad we are recording it so people can go back and reference it and all the rest of that. It's now my pleasure to introduce Dr. Aude Watrelot. And Aude already mentioned if you have questions, put them in the chat box or in the Q&A. We'll answer them all at the end of the webinar here. So a proper introduction of Aude. Aude Watrelot, Dr. Aude Watrelot is Assistant Professor of Enology in the Department of Food Science and Human Nutrition at Iowa State University. Dr. Watrelot's research area is fruit, grape, and wine tannin and polysaccharide chemistry and their relationship with wine sensory perception. Dr. Watrelot graduated with a master's degree in biology and health from the University of Nantes, France. Following this, she pursued her PhD studies at the French National Institute for Agricultural Research, INRA as it's known, and at the University of Avignon, also in France, where she obtained her PhD in food science. The focus of her PhD research was on the interaction mechanisms of polyphenols and pectins in a model system, the apple, i.e. for making cider. Following graduation, Dr. Watrelot decided to continue conducting research into polyphenol macromolecular interactions, but with a model change. And that's why she moved to California and conducted research on red wine chemistry with a specific focus on the mechanisms of astringency at California State University, Fresno, and at UC Davis, both in the Kennedy and Waterhouse laboratories. After 4 years in California and prior to working at Iowa State University, she moved back to France to conduct research on the chemistry of enological tannins at INRA, Superagro in Montpellier, in the Cheniere laboratory. And there you go, Aude. That's quite a background.
Good, yeah. Thank you.
Love to hear your part here.
Can you see my screen correctly?
Yes, we can. You can
okay.
Make it full screen, I think.
Oh, okay. Because I tried, but it's going on my second screen.
We can see the screen, but of course we also see the bars around it. That's a little better. Oh, now we see your desktop.
Oh, okay. Let me change that. It's coming.
I'm sure it is.
That's the thing when you use different screens. It's never working the way you want. Okay, how is it now?
There you go. Now you have it. You look great.
Okay, good. So probably you will see my face on the side more because I'm using the other screen. But anyway, you're gonna see the content. All right, so we will probably re-explain a little bit of things that Pete explained, but that just to make sure you are all aware of what are enological tannins, how they can be used, and why they should be used. So just a little bit about what does research tell us about enological tannin in winemaking. So the plans of today, that's really to explain you why tannin are important in winemaking, because that's really something you should consider when you make wine. And then where you can find different tannins, and you will see, and now you already know, I think, that you can find different type of tannins depending on their sources. And they will all have different profile and different effect on the wines. And then why could you use exogenous tannin or enological tannin? That's because they will have an impact on color stability, on antioxidation, or they are antioxidants, so they can help protecting your wine during aging, but also they bring some mouthfeel characteristics. And then we will just finish to summarize everything I mentioned. So the first reason to use tannin and why tannin in general are important in winemaking, that's because that's related to the wine quality. To get a wine quality, you need a balanced wine, meaning you shouldn't have one specific compound that will overpower everything else. So I did represent as a scale, but it's not exactly how it works. Or on two sides of the scale, you're going to have sugar or alcohol or sweetness, everything related to sweetness on one side, that should be balanced with other compounds. That's why I put the acidity in front of that, because when you have a high acidity, you will balance this acidity by adding a little bit of sugar. That's the same story for tannin. So tannin, that's also in the balance. And if you don't have enough tannin, you will have other compounds that will be overpowering your wine. So you need a little bit of tannin just to balance with alcohol, but also with acidity. And so the acids could also be on the side with sugar and alcohol. So that's really one of the main important reasons to have tannin in wines. That's to have balanced wine, high quality wine, but also that's in terms of perception, but that's also good in terms of preservation of the wine. So I just wanted to show you a study that has been done more than 11 years ago now, in 2010, that was on the relationship between the level of phenolic compounds and tannin, the concentration of those compounds, and how red wine were graded. And so you can see on the graph, so there is two graphs on the same figure. So that's for Vitis vinifera wines, red wines. On the left, that's for Shiraz. On the right, that's Cabernet Sauvignon. But you can see that the wine grade was always higher when you had a high concentration of tannin. So here, that's only showing high concentration. You can see that the concentration of tannin in those wines are up to 3 grams per liter. You will see that's not exactly what we have in terms of concentration in hybrid wines. But that's always something to keep in mind, is that red wine grades are based on the concentration of tannin and total phenolics, or phenolic compounds. So that's really important to understand the extraction of those compounds during the winemaking process. So extraction of tannin, when you make wine, that happens not exactly the first day of alcoholic fermentation. So in grapes, you have many different compounds, many different types of phenolic compounds that includes anthocyanin. So as Pete explained, anthocyanin, that's really related to the color, the red color of the grape skins, also grape flesh sometimes, and red wine. And so that's the main compound that will be extracted during the first days of fermentation. That's just because they are more aqueous soluble. And so when you don't have alcohol formed yet, you will start by extracting those anthocyanin. Then tannin will start to be extracted a little bit later than that, just because you will need to have a little bit of alcohol produced to help extracting those tannin. And so the first tannin that's going to be extracted would be more the skins tannins and then the seeds tannins. Just because the structure of skins versus seeds are different, they don't have exactly the same type of compound. And so that would need a little bit more time to extract the tannin from the seeds because they have lipids, they have a really hard structure. So you can see that at the same time you have extraction of anthocyanin first and then tannin, you also have extraction of other compounds that includes flavonols, which is another type of phenolic compound, but also you will have extraction of polysaccharides. And that's important to know that because you will have formation of bonds between tannin, anthocyanin, polysaccharides, proteins, all different compounds during the winemaking process. And so to extract tannin, because as you know now, that's very important for the wine quality, red wine quality. So winemakers and you probably already started to use different techniques. And so some techniques have been listed here just to maximize the extraction of tannin. That can include some stem inclusion, where in the stem of the clusters, you have some tannin into this part. That's mainly hydrolysable tannin, so it's not exactly the same tannin that you can have in grapes, but that's a way to extract more tannin overall into your wine and bring up the concentration of tannin you will have. Also, you can use saignée treatment, where that's gonna play on the concentration of your juice, and so concentration of your compounds found in the wine. Also, temperature of fermentation can play a role. If you want to extract more anthocyanin, you will tend to use cold temperature for a few days before starting to have the beginning of alcoholic fermentation. Then, if you want to extract tannins, you may use high temperature for a short amount of time. Because you don't want to cook your berries, but you just want to extract a little bit more tannin. Also, you can use enzymes, and that's similar to what Pete explained. That's to help degrading some of the cell wall material or the proteins that you have in the berries, and that could help extracting more tannins. Also, cap management. So that's something really important to extract more tannins. So when you have alcoholic fermentation, you have the formation of cap. And so this cap, if it's not constantly in contact with juice or wine, that won't help extracting tannin. So that's always good to keep this cap in solution or like wet, just to improve the extraction. And so to do so, that's one of the reasons why we do punch down, or that can be pump over. That's to help extracting more. Also, extended maceration, that can be helpful to extract more tannin from seeds. So that's a slightly different type of tannin, but that can help to improve the final red wine quality. And also you can use other new technologies like flash détente that use temperature, or high hydrostatic pressure, or ultrasounds, irradiation. Different techniques has been developed. And so regarding to extraction, I just wanted to show you, and probably you already saw that graph or that manuscript that has been published. So that's to show the level of tannin that are found in grapes and then the level of tannin found in wine. So that's following the regular or regular normal winemaking process. And so on the graph on the left, you have the concentration of tannin in berries from different varieties. So here, I hope you can see my mouse. On the right side of the left graph, you see the Vitis vinifera grapes. You can see you have a good amount, I would say, of tannin either from skin or from seeds in the grapes. In interspecific hybrids or French American varieties, you have a little bit less tannin content in those berries. But then when you look at what you have in the wine from the same varieties, you can see the level of tannin in Vitis vinifera. It's still pretty high. It's pretty good. But then for French American hybrid, that's not the same story. You don't have a lot of tannin. You are really low, and it's about 15-fold less tannin that you have in French American hybrid wines compared to Vitis vinifera. So that's one of the reasons why with hybrids, we would like to improve the way of getting more tannin. We really need to have more tannin in those wines. So some winemakers, we use techniques I previously mentioned. Otherwise, you can try to add enological tannin. But then, as we discussed with Pete, what type of tannin should you use and when to use them and how much should you use? So just briefly about enological tannin, what they are. So there is a definition, meaning that enological tannin, they are commonly used in winemaking to help stabilizing color and facilitating the clarification of must and wines, but they must not modify the color, the wine color, neither bring further aroma. So that's the definition from the International Enological Codex. And also, as previously mentioned, tannin have been discovered during the production of leather, where those tannin coming from plants were able to interact with the proteins from the collagen that was in animal skins. And so that's exactly what Pete explained. That's really tannin are everything that are able to bind with proteins or polysaccharides or alkaloids. So as Pete explained, so that's a little bit of repetition, but at least you're going to be able to know by the end of today what they are. So there are two types of tannin. So you have hydrolysable tannin on one side, which are non-flavonoids, meaning they have this structure. So you have a sugar and then different gallic acid connected, which represents ellagic acids. So that's called ellagitannins, and here that's a gallotannin. Those hydrolysable tannin are found mainly in wood, so in acacia trees or bark, in gannat trees, in oak wood, and also in some tara or quebracho, so that's a quebracho tree. On the other side, you have flavonoids, which is more including condensed tannin. And so you can see they don't have the same structure. Condensed tannin, that's just polymerization of different monomers. Here you have one monomer that is connected to multiple other monomers. So condensed tannin, they are mainly from fruits. So grapes, but also apples. And also you can find those type of tannin into quebracho tree. So sometimes, even if it's coming from wood, you also have condensed tannin. So that's just to show you the differences between those two. So as previously mentioned, I'm not gonna spend too much time on that. Enological tannin that can be helpful for color stability. That's because depending on their structure, they will be able to interact with anthocyanins, the color, to form new compounds, new pigments. Anthocyanin, as they are, right after extraction, they are not stable over time. So they can react really easily and really fast with other compounds. Once you have this new compound that is formed, the color is slightly different, but it's more stable over time. So that's one of the goals when you use enological tannin. That's to make sure you will form new compounds, new pigments that will be stable over time. Sorry. Another reason to use enological tannin, that's to protect wine against oxidation. So here that just, you don't need to know all about this figure, but that's just to show you how oxidation can happen. And tannin, when you will add them, that will play on those reactions. And so tannin can bind with metals like iron. And so when you bind with iron, you will just chelate those reactivity, and that will be protecting wine against oxidation. That will play on those different steps of oxidation. Also, that those tannins can, as I mentioned, can bind with proteins. Enzymes are proteins. And so tannin can interact with enzymes and to inactivate those enzymes. So that's another way to protect wine against oxidation using enological tannin. Also, as I just said, tannin can bind with proteins. In our saliva, we have proteins. And so when you have tannin that binds with proteins, you're forming some complexes and then aggregates, as you can see on the pictures. And those aggregates, that's what is formed in your mouth. Then you just swallow that. And those aggregates are leading to a loss of lubrication in your mouth. That's what we have as the astringency perception, the drying sensation. That's due to those interactions. So now I just want to show you some results about research that have been done about enological tannin. So in one study in 2018, so not many years ago, commercial tannins, commercial products were analyzed. So 36 products were analyzed. That was including grapes, grape skins, grape seeds, oak, tara, chestnut, gannuts, a lot of different type of enological tannin. So that was a mix between hydrolyzable tannin and condensed tannin. And so the first thing is looking at the actual content of tannin in those products. And depending on the type of product that was characterized, the actual concentration of tannin was about 13% in the oak or chestnut product. You can see that's really small amount of actual tannin in the commercial product. Then the same thing was done in the grape skins and grape seeds commercial products. In those products, they obtain about 72% of actual tannin. So that's just to show you how much real tannin or reactive tannin are available in the commercial enological tannin you may buy. In the same study, they tried to evaluate how much was the oxygen consumption rate for different type of enological tannin with different concentration used into the wine. They evaluate what happened for skins or grape skins versus oak enological tannin, and they observed that when they increased the amount of enological tannin added to the wine, it was just increasing the oxygen consumption rate, meaning that it was more protective against oxidation. With the more enological tannin you add, the more protection you get, which makes sense. And then they did compare what they have between grape skins and oak. They observed that oak was leading to a higher oxygen consumption rate, meaning that it was more protective against oxidation. And that's because oak, if you do remember, that's a wood, and so it's containing more hydrolyzable tannin compared to condensed tannin. And so the elagi tannin, so one type of hydrolyzable tannin found in oak, was more protective against oxidation because it reacted a little bit stronger with oxygen or metals. So once again, what is really in enological tannin? That's always the main question, because you don't want to add too much enological tannin during your winemaking if you don't have real effect, right? So you need to know exactly what you're adding. And in a study that was done in 2012, and in one of the study I did at INRA Montpellier, Supergro Montpellier, was to look at how much actual tannin was in different products, hydrolyzable enological tannin versus mixed tannin. And so in one of the studies, they observed that it was containing only 12% or 48% of actual tannin in the different products. So you can see the hydrolyzable tannin contain a little bit of actual tannin, but it was only representing 12%. And the other mixed tannin represented about 48%. So it's still pretty low. But then it depends on the type of enological tannin you evaluate, because in one of my studies, I looked at the actual level of tannin from a gallnut enological product. And the actual concentration was about 86%. So it was pretty good number, but that's really depending on the type of product you get and the type of method you use to characterize those concentration. So in the same study from 2012 by Harberton, they evaluate the addition of different level of enological tannin in a Merlot, so vintage vinifera wine. They observed by adding from no addition to 300 milligrams per liter of enological tannin into the properties of the Merlot wines, they didn't observe any effect on anthocyanin content. They didn't observe any effect on small pigments or large pigments, polymeric pigments. They observed a slight improvement of the tannin concentration, but it wasn't that crazy and nothing really beneficial, I would say, for the quality of these wines. So they did try to add a little bit more of the enological product, enological tannin. So they increased to about 800 milligrams per liter, which is the, I think, legal limit of addition. And they did observe an improvement of tannin concentration. However, the sensory perception, so aroma compounds, the aroma profile, was negatively impacted. So it really depends. Always try to keep in mind that depending on what you're going to add, what type of enological you're going to add, maybe you will lead to sensory impact, negative sensory impact. So now, just to compare between two studies that were done in 2013 and 2016 on Maréchal Foch, Coro Noir, and Lamberger, which is not a hybrid, on the addition of about 40 grams per hectoliter of enological grape tannin, so condensed tannin. The same study was done on still Maréchal Foch, but with a little bit more of those enological tannin. They did look at the concentration of tannin after those addition. Between the control and tannin addition, in Maréchal Foch and Coro Noir in 2013 seemed to be improved compared to the control. However, when you look at the study in 2016, they did observe a decrease of the level of proteins in Maréchal Foch, but they didn't observe any changes in the level of tannin. So that just to show you that depending on the varieties you use, you may have differences in terms of impact of enological tannin. Also, depending on the level of addition, you may have a little impact, but maybe you won't have an impact. And also it depends on just what compounds, other compounds you already have in your grapes or in your wine. Depending on the level of anthocyanin, the type of anthocyanin, you may have improvement or not of the concentration of tannin. Depending on the level of polysaccharides, depending on proteins, a lot of factors will have an impact on the addition of enological tannin. And then the last study in 2019, so that's a little bit earlier, they did look at the impact of commercial tannin, so that was grape skins or seeds that actually contain 38% of actual tannin. So they did use different concentrations. So they did add 400, 800, and 1,200 milligrams per liter, which is really high concentration So they did add those different concentration in 3 different wine. In Maréchal Foch, on the A figure, B is Coro Noir, and then Vitis vinifera Cabernet Franc on the C. And so then they did compare the level of tannin over time during all the winemaking process. They did observe that by applying a high concentration of enological tannin, they did improve the concentration of tannin. For all of those wines, you can see the 400 milligram per liter is really low in tannin concentration compared to 1200 milligrams per liter. But then if you look what happens along the winemaking process, throughout the winemaking process, you can see that the retention of tannin is not exactly the same. For Cabernet Franc, you have a little bit of retention of tannin. So the level of tannin is quite constant over time. You have about 34 to 48% of retention of tannin. But then when you look at Maréchal Foch or Coro Noir, you see a decrease over time of the level of tannin, meaning that you don't have a good retention of those tannin over time. So that's really something to keep in mind. That's not because you're going to add a bunch of tannin, enological tannin, to your wine. That means those tannin will stay and will still be doing their job. So it really depends, as I mentioned at the beginning, or in the previous slide, it really depends on the variety you have and what other compounds are already in your wines. And so, yeah, I tried to make it quick. I hope it wasn't too fast. Feel free to rewatch the recording. But just take home messages. So the wines from hybrid varieties, that can be beneficial to use enological tannin, but you should know exactly what type of enological tannin you want to add, for what reason, and when to add them. Really think about the purpose of addition of enological tannin. Also, it always depends on the varieties, and it's also depending on the viticultural practices. It's not because you're going to have market wine in Wisconsin that you will add one specific enological tannin. That's going to probably work on your market wine in Wisconsin, but probably it's not going to work on market from Iowa, because you never know how is the grape, the chemistry of the grapes initially. Also, the commercial tannin, keep in mind, it doesn't contain 100% of actual tannin. So maybe you will need to add a bunch of enological product to make sure you have the appropriate amount of tannin that will be reactive. Then also tannin, they interact with different compounds, especially nitrogenous compounds, so proteins. So that's why it can have a negative impact on the level of tannin you have in wines. And something I didn't really mention, but Pete explained that a little bit, that's about the relationship between tannin and anthocyanin. So to get enough, or to get a good red wine quality, you would need a good ratio of anthocyanin or tannin over anthocyanin. Usually, a good ratio should be between 1 to 4, meaning you should have about more tannin than anthocyanin. So in Vitis vinifera, we tend to have about 500 milligrams per liter of anthocyanin and 1 to 3 grams per liter of tannin. For hybrids, that's the opposite that we have. So you can see there is something that we still need to figure out, how to reduce a little bit those anthocyanin, improve the tannin concentration to make sure we have stable wine color and stable wine quality. And so then also, if enological tannin, you can use enological tannin, but you can also try to extract tannin in other ways or do combination of different methods. That's all I have. So if you have any question, feel free to send me an email or just go on my website and you will have some information about characterization of phenolic compounds and tannin.
All right. Thank you, Aude. That's
Yeah.
a lot of information there.
Yes.
We already have some questions in the Q&A box or the chat box, which I'll read. But we're only left with about 15 minutes to get through these, which might just be enough. I'll go ahead and read. This is from Patrick Pirquet, and this is to both of you, or to me, I suppose, as well. And you covered some of this in your last slide or two, Aude. Patrick asks, I had heard from other researchers that commercial tannins are at most 50% actual tannin. The rest is other material. Does this other material make a positive contribution to the wine? And if not, have any companies devised a way to make a product that is closer to 100% tannin? Or is this just impossible to do in the processing?
So I can start on that just because I did mention it a little bit. And we had that discussion with Pete. Yesterday we did a short training and we discussed about it. So as I presented, some of the enological products, they only contain, as Patrick said, only like 40% or 50% of actual tannin. But it depends also on the way of characterizing those tannin concentration. Depending on the method that is used, sometimes you will have a low percentage, sometimes you may have a high percentage. It depends on how those characterization has been done. But most of the time you also have other material, and those other materials that can include some polysaccharides, like cell wall material, or that can include probably proteins. And maybe, Pete, you have more information about what are those other materials. Because true, I'm sure that's also something that can react with everything else in your wine. So it's not only the tannin that you will be looking at or the effect of tannin that you will add, but also the other material you will have into this enological product.
Right. Well, to address the first part of what you said, I think also to measure the amount of tannin, it depends on the assay you're using and how you prepare your sample. If you're measuring monomers, then you do the fluoroglucinolysis or whatever, you do different ways to prepare the sample so you can measure what you can measure. Saying there's a certain percentage, you really should always have to say the type of sample prep and analysis that you did. Because when you extract these complex molecules that are different sizes from a complex matrix, like say oak, you may be pulling out large polymers of these things that don't get quantitated in the sample prep, but still certainly have an effect when you add them into the wine. It's a complicated matrix. It's much like saying, can you define every aspect that's in a wine? And oh, by the way, each of the molecular interactions between them as well. The answer is no. We don't have the technology to be able to pull things apart that way. It's just too complicated. I would say one thing, though, about the question of making a product closer to 100%. Can it be done? I'm absolutely certain it can be done. The question is, would you pay for that product? Because it's going to be expensive. Sometimes you're better off to buy a product that seemingly has less of your active compound, but costs maybe 1/10 of what it would cost. Say you get a 50% tannin product. If you made 100%, it would cost 10 times as much. So, there's always trade-offs commercially that you look at in that instance.
Excellent reply from both of you. I'm just going to remind everybody, with grapes, with French hybrid grapes, they've been around quite a bit longer, but specifically the Minnesota varieties, these grapes still haven't even been around a generation yet. The research, I can assure you, is ongoing. I mentioned earlier before we started that Aude and I have done a research project, a cooperative project last harvest on the use of partial whole cluster fermentation in Marquette. And so we're curious about the same thing that all of the Midwest winemakers are curious about, and we're working on it. And thank you all for your participation and for asking such good questions. There's another question here from Patrick Pirquet. Sorry, Patrick, if it's Pirquet. Pete, you focused on commercial tannin addition for red wines. Do the same principles apply to orange wines? so-called orange wines. Do you have any idea on that?
Well, I have an idea on about everything, and they're worth about the same amount. But I think, I'm not overly familiar with orange wines, but I am somewhat familiar with the chemical aspects of the pigmentation of anthocyanins that lean towards the orange color versus, say, red or violet and blue. And it can be pH effects, but can also be chemical structural effects. So just with that bit of a preface of my limited knowledge on that, my answer would be, yeah, I think that the principles we spoke about apply as well. You're just dealing with anthocyanins that bring a different color, but they have the basic structure that is the same, and stabilization of the electronic center that does the wavelength conversion and emits the wavelength of the orange color is going to be very similar. And so again, trying to build the stability in red wines is going to be very similar to building. It's like a challenge. Here's a similar challenge. Rosé. The way rosé wines are made, there's you press off. There's not a lot. The tannin anthocyanin ratio is very off. It's more like hybrid varietals than it is, say, red wines. Okay, so stabilization of color in rosés is a bit of a challenge. And I know we've done things in the past, like the use of carboxymethylcellulose actually did help stabilize color in rosé wines, as well as, of course, it's a tartaric acid stabilization treatment as well. And now there's the polyaspartate. I don't know what polyaspartate does with the colors in rosés, but it also is again a tartaric acid stabilization factor. So sometimes things that you add to wines for one purpose have effects in other areas too. So even though I was pretty heavy on the chemical interaction and the molecular bonding of the phenolics to protect the color center, there's other molecules that can actually help do that too.
Yeah, I agree. I think rosé wine, that's really a good example of red wine that we can have as hybrids, where that's really tricky to stabilize just the color of rosé wine because they don't have tannin. You don't extract those tannin. Yeah. Let me just ask the question that Bill asked me right before my presentation. So he's a hobby winemaker. I have a sleeve of 6 different enological tannins from BSG. Are you familiar enough with their product line to advise on which might be beneficial to market? And I would say that depends on the type of purpose or the purpose of your wine for market. Do you have any ideas, Pete?
Well, I didn't get a chance to mention this, but I did some collaborative winemaking trials with some folks in New York, Ontario, and Missouri. And one of the wines we worked with was Marquette. And what I did is I did actually VR color and a VR supra, so the fermentation tannin, enzyme tannin treatment on it. And it responded slightly, not the way we had hoped. And of course, some of the things we talked about is the reason why. Again, I think bench trialing is your best bet to try to figure something out. If there was hard and fast rules, I'm sure BSG wouldn't have given you a sleeve of 6 samples. They would have given you 1 because they knew that that's what the answer was. They don't know the answer, and neither do you until you try it.
The research is ongoing. Quick question here. Can you increase tannin in red wine by adding pressed pomace from another red wine? And I'm just going to say real quick, I'm not so sure about adding pomace from pressed fermented wine. In my Pinot Noir days in Santa Barbara, a few winemakers would add the pressed unfermented pomace of Pinot Gris, of a white grape or a gray grape, to their Pinot Noir fermentation, and part of that was for some of the tannins in the ends, and that's as far as I'm going to answer that. Do you have something to add to that, Pete?
Oh, let's put some light on the subject. There's so many additions that I've seen, especially used with Pinot Noir to try to build a little more. I've even seen some like post-fermentation Syrah some of the skins, because you don't get a whole lot of color and you can still get a little bit of structure out of that. Of course, there's the, you know, aging stems and then adding those back in as well. I would imagine, depending on the pomace itself and how highly extracted it was, did you go through a long pre-fermentation maceration? What you evaluate what's left in it, and also the condition of the pomace, because sometimes it can get a little nasty microbiologically. Those are the kinds of things I think about. But could you gain some characteristics by adding that? Sure. Could you gain characteristics by just taking the pomace, making a slurry of it, okay, and then maybe filtering it and adding that back to your wine instead of dumping the whole pomace in? Yeah.
Yeah, I would say you could use the pomace, but as Drew, you mentioned, I think that's better to use the unfermented pomace, just because the tannins are still in this unfermented pomace. So you can still add that back to your wine and extract more of the tannin from those unfermented pomace. But then making a slurry of the pomace, I don't know if you would still be able to extract those tannins before adding the slurry.
I think that comes from my Gallo days, where there's no unused part.
Yeah.
and if there is something laying around somewhere, you try to find some process you can do with it to get something else usable out of it. Just, but you know, when you have those kinds of resources and the different product lines that are available to dump stuff into, those kinds of thoughts come to you.
But then that's also a good question to reuse pomace, just to get a little bit of sustainability, just to make sure you use everything from your winemaking and to avoid losing too much of your winemaking thing. That's always a good way to add value to a waste, you know?
That's a wonderful point. The Europeans, of course, in my experience, the Italians in particular, of course, will always use the pomace to make grappa or a marc or piquette. So I love the idea of reusing for that purpose. I don't want to spill anybody's secrets, so I'm just going to say I know a winemaker in Minnesota who has been taking the pomace of a certain white grape and adding it to their fermentation of a certain red grape. And that's all I'm going to say about it. So again, the research and experimentation continues. We've got just a couple more questions here. We've got 3 minutes or so.
Drew, if I could just mention one more thing about the
4:30.
Sorry, Drew, I don't mean to talk over you. I really apologize for that. But I wanted to add one more thing about the pomace, and that's as a winemaker, if you want to try that, keep in mind, I mean, if it's a white, you're going to press it off. It's going to be what it is. If you're going to use from a fermented red, keep in mind that you may want to do that from the beginning. And the question about the submerged cap that is in the chat box, you know, understanding that you want to use that pomace later, maybe you want to keep that cap extra wet. Maybe you want to keep the surface of it from becoming too colonized with mold and bacteria that you might not want later if you're going to reuse it. So having, you know, if you have the plan ahead of time, you can prep the material a little bit to eliminate some of the potential downside.
Fair enough. Here's a question from Bob Thaden up in Montana. And he's asking, how do Marquette, Petit Pearl, Crimson Pearl compare to Noiret or Noirette and Coral Noir and Pinot Noir in tannins? I can tell you that Pinot Noir, being vinifera, has more tannins than any of the hybrids. Petit Pearl and Crimson Pearl are notable that they do have a higher amount of tannin that survives through fermentation. Marquette, we know, has high protein, and unless you add some tannins, it can end up on the lighter side of the tannin scale. As for Noirette and Coral Noir, I haven't a lot of experience with either of those. Again.
And I would add also something is that when Bob, you're just asking about the level of tannin, but also think about other compounds that are different in those varieties. So Marquette and Pinot Noir tend to have similar profile because Marquette has a little bit of Pinot Noir in the background. But if you think about other compounds, so tannin, yes, Drew, you're right. Petit Pearl, Crimson Pearl, it has a little bit more than in Marquette. But then the level and the type of anthocyanin is also different. So when we look at the concentration of tannin, we always try to look at tannin overall, but we don't really consider what are the level of tannin after interaction with anthocyanin. And we know that anthocyanin, depending on their structure, they would be more or less able to form new pigments. And so that's also something to keep in mind. Don't look only at the level of tannin available in those varieties, but they also have different anthocyanin profiles. And so the level of tannin can be a little bit different just because of that. I hope it's clear.
Bob, you always know where to find me or Aude afterwards for follow-up. We've got one more question in the chat box that I can see. And I think we're just at 4:30, so we'll make this the last question.
I have another one at the end.
Oh, you do? Okay. Well, this question is, can you explain about the use of sacrificial tannins and how that works again? Can one of you do that briefly?
Sure, let me take a swing at that. Adding a sacrificial tannin very early in the fermentation process will occupy binding sites in the wine must that your skin tannins would attach to if it was available. So when you add that sacrificial tannin early, it takes the place of a grape tannin that may be taken out of solution by binding to the matrix.
Good.
All right. And you had one more question that you saw?
Yeah, I did have one from Bill. Okay, let me find it. Okay. Also, can you discuss the use of oak chips in fermentation or cellaring versus enological tannin additives or a blend of the two? Pete, you want to answer that? Or
sure, I can talk about it a little bit. Well, let's see. First of all, let's just talk about oak chips just for a second. One of the benefits of oak chips is that you get those and they're processed. They can be raw or they can be toasted, light, medium, heavy, and you can get flavor aroma compounds out of those chips that in enological tannin additions you usually don't get unless, finishing tannins, yes, because you're using the same thing basically. You're toasting the oak and then you're extracting those compounds from it and adding it back in very small amounts of a soluble powder. When you add the oak chips, you can get an extraction of those same compounds depending on the toast level and the production and also the source of the oak, much like I'm not going to get into barrel selection and oak differences, but of course the compounds that are produced from American oak, French oak, Eastern European oak, they differ. Oregon oak, but you also get the hydrolyzable tannin extraction when you use oak chips. It's a slow extraction, so reactivities are not pushed. I don't know what else to say about it other than that basically finishing tannins are oak chip flavors without the solids, because it's a soluble port. Most everything in the powder at the end is soluble into the wine, so you're gaining the flavor. And early on with the chips, you can get some of the flavor contribution, but you also get the hydrolyzable tannin.
Yep, all good. And I still have one last question. Sorry. Any comments on pressing reds, protein fining the juice, then reintroducing it as opposed to adding sacrificial tannin? Is this just a ton of work, or will it make the tannin addition more successful long term?
Personally, I think that protein fining the red wine would be pretty detrimental on a number of factors. I think that you're going to pull out some flavor aroma compounds as well. And anytime you do a fining like that, where say like I'm imagining you're saying protein fining, you want to throw some bentonite in there or something like that. I'm not in favor of that very early on. In some extreme cases where we've had compromised fruit, we've fermented on bentonite for white wines. But for the most part, throwing it into red must would create quite a mess, I believe.
Yeah, I think this question, Pete, refers to a technique that's been investigated, I think by Anna Catherine Mansfield, of taking, let's say, Marquette or Frontenac, two University of Minnesota red grapes, pressing them off, retaining the pomace, I guess in a chiller or a freezer, adding bentonite to the pressed juice, allowing that a couple days to do its work, racking that clear juice off of the bentonite, i.e. without proteins, and then putting that back with the pressed unfermented pomace, and then going through a normal fermentation. I can tell you, I know people have done it on a small scale and they were relatively happy with it. I think on a large scale, I think trying to do that with anything more than about 2 tons would take some serious extra work. As I say, you're going to have to keep those unfermented pomaces in good condition. The 2 or 3 or 4 days you're going to need to let the bentonite do its work on the pressed juice. So try it. If you like it, great. If I had to do that with 20 tons of Marquette, I think I would leave winemaking.
That's a good final comment, Drew. Thank you. I think we are all done with those questions. If you have more questions, feel free to send us an email and we will direct the question or we will answer you. So thank you very much to all of you for attending, and to you, Pete, to be with us today and to present on Enological Tannins. Thanks, Drew, for helping me for this organization and co-hosting the webinar. So that was the last of the year, but hopefully we will provide you more next year on different topics. So please take a few minutes to take the survey, to complete the survey. I will probably send you another email with the link with the survey, just to make sure you can write any suggestion you have. And otherwise, I would say thanks very much again for attending, and we will see you soon. Happy holidays, and see you next time.
Take care.
Thanks, everyone.
Happy holidays to you all.
Yep.
Take care, Pete. Thanks for the help.
Thanks for having me. I